Dear students, in this module, I'm going to talk about the strategies that you can employ for comparing the protein structures. As you know that the PDB files, they contain the alpha carbon coordinates that are there in the backbone of a protein. And you can compare the alpha carbons in one protein to the alpha carbons in the other proteins, and then see how similar these two proteins are. More so, you can compare the entire proteins or just portions within the two proteins. The portions can be motifs or domains. So it depends on if you're trying to do a whole protein structure comparison or just a partial structure comparison. In this figure, you can see that you have the backbone of a protein shown here, and you have the backbone of another protein shown on the right side. So if you wanted to compare the alpha carbon positions that are indicated by these corners, so what you should do is, you should find the corresponding alpha carbons in the backbone and then compare them with each other. So assuming that one protein has 10 alpha carbons while the other protein which you are trying to compare has just 9, then it can create a problem. So to overcome this problem, you have to be very careful, and you may want to have a well-defined strategy for that. So here I have outlined important steps in such a strategy to compare the whole protein structure. So this will help you to solve the problem that I just mentioned, that is, that if the protein you're trying to compare has a different number of alpha carbons to the one that is unknown. So the first step in this is that you have to do pairwise sequence alignment. So if you do pairwise sequence alignment, then you will be able to see which alpha carbons correspond to which alpha carbons in the other protein. Of course, there may be gaps, mismatches, so you can ignore those amino acids or the alpha carbons that are involved in those amino acids. Secondly, so once you have done that, then you look at the corresponding alpha carbons and you extract their coordinates from the PDB. Now you have extracted the coordinates from the PDB for each alpha carbon that corresponds to each other. The next step is to calculate their difference. And to calculate their difference, you can simply use geometry and you can arrive at their distance. I'll talk about this in the later modules in detail. Also, so once you have calculated their distance, then you may want to talk about the quality. So to talk about the quality, there is something called as the root mean square distance, or simply the RMSD. So the RMSD tells you the quality of comparison between the two protein structures. If the RMSD is big, then the two proteins are different. If RMSD is small, then the proteins are similar in structure. In this example, I will talk about two proteins, the blue and red one shown here with triangles. So the first step in our strategy was to compare the alpha carbons and see which ones are corresponding to each other. So in this case, alpha carbon 1 corresponds to alpha carbon 1 on this, 2 corresponds to 2 here, and 3 corresponds to 3 at this protein. Now, since you have computed which alpha carbons correspond to each other, then you may want to do the distance between 1 here and here. So this one is indicated in blue. So you may want to extract the coordinates from the PDB like that and compare them with these. Thus you will be able to compute the distance between these two. And you can do this for all these different alpha carbons as well. Thus you will calculate the distance between the overall structure of the protein. Now once you have done that, the root mean square distance needs to be calculated. So this is very important because it is going to summarize your entire comparison. So RMSD, as I mentioned earlier, if it is small, then the structures are similar. If the RMST is large, it means that the two structures are not much similar.